Yeah, we can see you and we can hear you, but it's not your full screen. You are presenting just.
Can you see
please
now?
try to make it full. No, nothing changes. You can press F5. You can try to do it.
I already did it. You don't see it?
No. Oh, now it's something was downloading a few seconds ago. So. Oh, just a black screen now instead of the slide. So maybe we can just look at the smaller version.
Okay, sorry.
It's absolutely normal.
Good morning.
But now we cannot see your presentation. Could you please come back to the previous version? This one, but probably you need to change the slide manually, like to go down to just make any changes.
Can you see now?
Yeah, this way. Yeah,
okay.
Yeah.
Okay. Well, good morning. I'm very happy to be here another year. And last year I presented something about teams, but this time I'm going to tell you about establishing the key factors for successful mussel biobanking. So in this work, we use the mussel Mytilus provincialis, which has a great economic interest. And we wanted to use a crab observation protocol already designed for mussels on different pre-selected mussel families to see if these families survive better or not to crab observation compared to the control. And the families we used were selected either by size, by second generation of big mussels, mussels that were exposed last year to extreme temperature shocks and have been cultured and survived for the last year. And also we pick a general sample for the local population. And the other objective was to test 3 new CPAs that were never used on mussels before, which were methanol and dimethylformamide and Cristor on mussel eggs. So we collect the gametes and did the fertilization to obtain embryos that were incubated at 18 degrees for 72 hours to finally obtain the larvae that were cryopreserved. For cryopreservation, we use 15% DMSO final concentration added to the eggs, to the larvae, in one step, and they were incubated for 60 minutes to achieve equilibration. And the cryopreservation protocol consisted in holding at 4 degrees for 2 minutes, and then cooling at 1 degree per minute until minus 12 degrees when the seeding point was done. And after this seeding point, we continue cooling to minus 35 degrees to finally store the larvae in liquid nitrogen. And for thawing, we use the immersion water bath at 35 degrees for 6 seconds. After cryopreservation, if the larvae develop to juveniles. On the other hand, for toxicity tests, we use from 0.5 molar to 3 molar of methanol and dimethylformamide. And we also use an usual cryopreservation media called Cristor, which we know that it has 10% of DMSO, but the rest of the composition is patent and we don't know it. And for this CPA, we use from concentrations from 5% to 50%, and all the CPAs were added in 1.1 radio and in one step. So we expose the eggs to these cryoprotectants for 15 minutes. Then we wash out them and transfer to filter seawater. After that, we fertilize and incubate them for 72 hours to see the percentage of development until the larvae stage. So we found that after 2 weeks for postdoc, the survival drops compared to the controls of every family, and the best results were obtained with the general sample from the local population, and the worst ones was with the FB1 family, the one that is comes from the second generation of pig mussels. On the other side, if we look at the percentage of settlement, we saw that it was higher in the crab self mussels, and the best results were obtained with the parental big family. Regarding the toxicity test, we observed that until 1.5 molar, we could use methanol or N-dimethyl-formamide. But if we higher up the concentrations of methanol, the percentage of normal larvae drops. And the other sepia tested, the cryostore, we saw that did not cause a big decrease, since 70% of the normal larvae was reached when we used the CPA, even at the highest concentration. So our results agree with those obtained before with this crab preservation protocol. For example, Rodriguez Ribeiro have found that less than 1% of these larvae coming from crab preserved Troncoso reach settlement, while Els et al. Found that with 72 larvae, which is the same as we use here in our experiment, 20% survival was achieved. And we also found that settlement was higher in the crab preserved group than in the controls. And we also saw that gamete quality also are affecting crab preservation results, as best gamete quality in our study was achieved by general sample group, and it had the highest survival after 2 weeks postdoc. And regarding toxicity tests, the use of these new protectants does not seem to greatly affect the development of the larvae, but we don't know if they will work with crab preservation. We don't know how they behave in crab preservation and if they are capable of protecting the cells in this process, and also if these cells also develop after thawing. And thank you very much for your attention.
And thank you very much for your talk. It was indeed informative. So I would like just to ask you, could you just provide us some recommendation for your opinion for the best techniques for crab preservation of mussels? What do you think? What should be the most suitable development stage, the most suitable protocols and so on? Just your opinion, not the rules, of course, because nobody knows.
Well, we saw that this protocol really works with 72 hours the larvae. If you want to crab preserve Troncoso or 48 hours larvae, you will find that survival is less than when we use the 72 hours larvae.
Thank you so much. It's indeed a good work that was presented. Do we have any other questions from the audience? To Sarah? And probably I also have another one about cryostore. Actually, why do you think that why the toxicity of the cryostore is like the less compared to all other types of crab protectants that you use? Could you please clarify what can be behind those data?
Well, the cryostore was the first time that we use it. We never used it before with any kind of cells of any kind of species. So we found that interesting because we could higher up the concentration until 50% of cryostore and 50% diluted in water. But we only know that has 10% of DMSO, the rest of the components, we don't know it because it's patent. So that could be if this CPA can protect the cells on crab preservation, that would be a really good component to use. We need to look forward on that part of the experiment.
Yeah, because maybe all of us also need to switch to the cryostore and cryopreserve everything with the cryostore as well.
Maybe
so thank you so much.
Yes.
Yeah, true. And now we have another question from Barry, please. We cannot hear you, so you probably need to turn on.
There we go.
You can't hear me. Thank you. Yes.
No,
yes. Great presentation. Just a comment, really, I think, about the cryostore story. It's highly likely, I think, that there are sugars in that mixture. So you may well be able, in your studies, to test DMSO with some of the other sugars like trehalose or sucrose and get similar kinds of good results. So I think we can all develop our own ideas about the best cryoprotectants, but you don't have to have these commercially available solutions. I think you can match them. You will be able to match up with other additives to DMSO yourself. Okay, great. Thank you.
Thank you so much for these comments. And we have another question from Brian Grout. Would you see D. larvae as a better choice for biobanking rather than storing of gametes? So what is your choice? Do you prefer and do you think that the storage of larvae and cryopreservation of larvae is a better choice comparing to cryopreservation and storing of gametes?
Because the larvae survive better to cryopreservation, as we saw in our results. So we decide to biobank these 72 hours larvae.
So for you, it's a definite choice. Larvae are the best. And probably the last question for you from Anastasia.
Yeah, thank you for your presentation. I have a kind of a practical question. What is the process how then this cryopreserved larvae can be introduced into the ocean or into some pools firstly, and then to ocean? What is the whole procedure? Because they are small and then we have like big mussels.
Okay, so we incubate them for 2 weeks. That's the time they use to do the metamorphosis and change into little mussels that you already know, like the shells and everything. And then we put them in ropes so they can attach to them and continue growing. And after they already attached to the ropes, you can put them in the ocean to continue growing.
Thank you.